but overall,
you guys just submitted well. Good setup. If you need to grab up really fast.
Okay, I got a real quick statement. Today we're eating Foxy's Best Beef Hot Dogs. Is there anybody that doesn't enjoy?
Is there a vegetarian?
Anybody? I need to know. Nobody? All right, it's beef hot dogs.
Yay! Hot dogs!
I just want to say, beef hot dogs are not steak hot dogs. They're not like hot dogs.
Yeah.
Tube steak.
I know.
So there's any packages.
Well, I'm just not.
I was TCK last year. This is year 2, year 1.
Thank you. And I don't know if I heard.
Yeah, right. Well, I got an upper freezer and then I got a small chest. And I do half hour and then we make a point of eating and when we're done, just like I really it's good for me. But 3/4.
Okay, let's go ahead and head back to where we were so we can get our next speaker.
Yeah.
As much time as we possibly.
We're back in.
I need to learn that whistle.
All
I guess most everyone in here has had to use that whistle on their own class. So they're really.
Just stand there like a moron. And then ask, like, check these for nay or something. No, but it's the whip.
That's a good answer.
Okay, we're going to continue with our look at teaching with fungal and microbiological world in mind with another KU faculty member, which is kind of hurting my K-State alumni heart. So many great KU faculty members here today, but it's okay. We have our keynote speakers who make up for all of that. He just bleeds purple. But this is Dr. Ben Sykes. He will be talking to us today about plant endophytes, which is going to be great. So give me your attention and thanks a lot.
That is how blue this jacket is.
It's free.
Yeah, so I'm going to talk a little bit about this, a little bit different from what Peggy was talking about, and then I think this is a cool platform that you guys might be able to use to ask lots of, lots of different questions, and actually let students drive different kinds of research. I'm just going to talk briefly about who I am and what kind of research we do in my lab and give you an example. And then I'll talk about what plant endophytes are, because it's not always obvious why they might be interesting, how you actually culture them, I'll talk for a minute about what kind of experiments you might do in your labs and what you do with them. And then I'm just going to explain why that might be interesting to you guys. I've taken a little bit of effort to look at the next generation science standards and look at how experiments that you can do with them might fit into the particular elements of the life sciences stuff for high schools at least. We've done some other stuff for different grade levels as well. So I'm an assistant biology. I haven't been here that much longer than Peggy has. I've only been here for about 2 years. I'm an assistant professor of microbial ecology, which basically just means I study the interaction of microbes with each other, with plants, with other organisms, with their environment, lots of different ways that I study them. Like Peggy, I'm actually cross-appointed 50-50 between a KU department and the Kansas Biological Survey, whose mission is to serve Kansans and do research on biology across the state. So a major portion of that is to do outreach. So I think that's really central to a lot of the stuff I do. Apart from who I am as a scientist, I also have, I think, just about every representative of education across the spectrum. I have the smallest almost to go into elementary. I have 2 that are in the middle of elementary, and then 1 that's in middle school, and Ken said he's in high school already. So I have a vested interest in thinking about how Kansas teaches biology, because for quite a while I'll have a representation across the biological spectrum. So what kind of work do we do in my lab? We work in some pretty cool places. These are some pine savannas down at the Florida-Georgia border. We do some work in the Great Smoky Mountains National Park. Of course, we work here in Kansas. These are some prairies down south of Garnett in eastern Kansas that are not on the Flint Hills that somehow this poach's managed to survive. This is my graduate student digging up some cattails in reservoirs around Kansas. So we work in a lot of variety of different areas. And we work on a lot of different kinds of things. We look at crusts that can form right on top of really dry soils. We do look at rhizospheres, at the roots of plants and the soils that surround them. And then we, a lot of times we just go out and find cool fungi. We kind of find cool mushrooms, and that's what gets students excited. But our research doesn't often focus on the mushrooms alone. So these are some of the projects that we work on. I just wanted to give you guys a smattering of it. When somebody says, well, what do you work on? It's kind of hard to encapsulate all that. Microbial ecology is it, but we ask questions like, can soil fungi create feedbacks that affect the probability of future fires? We ask some things like what Peggy was talking about with specific, some different kinds of microbes and AM fungi. If we can add them, that increases the effectiveness of ecological restoration. Whether or not, when you get more stress, like as you move west in Kansas and there's less rain, whether or not that makes species form mutualisms or rely on them more. So we have a cool project that's going on right now with that with some undergrads. We look at the timing of arrival of species to a new ecosystem or one that's been heavily disturbed. Does that timing matter? Of course, we do stuff out at the Free State with Julie's classes. We've gone out there and looked at how, after her and Helen did the amazing work to put in different plant species, whether or not that's changing the microbial communities in the different treatments, and then whether or not those might feed back to change the restoration rate in the different treatments. So that's some of the research that we do, just like a smattering of it. I'm just going to give you one example and talk about the fire stuff for a minute so you can see maybe what some of the in-depth stuff looks like. So the idea is that there might be a potential for feedbacks that can occur between fire and fungi. In systems that burn a lot, pyrogenic ecosystems, we already know that there's plant species which can engineer fire. So they have traits that allow them to bounce back after fire, tools which are more flammable, but nobody's really looked at any other organisms except for the vegetation. But we do know things like fungi are way susceptible to fire, but they differ in their susceptibility, and that the fungi can have different effects on fuels. And so this is just kind of an illustration of it. You start out with one fungal community in an area that hasn't been burned for a while. Fire happens, and only some of those guys survive. Okay? And the post-fire environment looks very different than the pre-fire environment, and some fungi are able to live very well there. Thank you very much. So fire sweeps through, some of the fungi pop up, and in fact, we know already that there's some that fruit that form mushrooms right after fire.
Morels.
Right, morels are a great example. So there's whole itinerant groups that follow fires in national forests hunting for morels. So the post-fire adapted fungi, though, the idea of the feedback is that these post-fire adapted fungi might have distinct effects on fuels. They can have them directly in how they decompose those, decomposing them, or they might have them indirectly through their effects on plants. Okay, and so those shifts in fuels, those modifications, could then feed back to the probability of new fires. All right, and so what we studied, I'm just going to give you the punchline, is this side, how fire changes the fungal communities, and then the blue line here, whether or not those fungal communities differ in their ability to break down fuels, new fuels. And all this work was done, we've tried to set some up here out west as well, and the Welder Prairie, but we've done this work so far at the border between Florida and Georgia at a place called Tall Timbers Research Station right outside of Tallahassee. It looks kind of like this. It's very blade-like and savanna-y. This is what it looks like when it hasn't burned for only maybe a year or two. Fire sweeps through, you can see this one is still burning, and this is actually only about a month after the fire has been there. So you can see how rapidly, all the plants are adapted to spring back after fire, and they do prescribe burns there. So this is a kind of an ideal system to think about other organisms like fungi that might be adapted to fire and able to spring back. So here's the punchline. From the first step, this is an ordination. Points that are closer together just means the communities there were more similar. And this is looking at whether or not fire shifted the fungal community. So the closed circles are soil samples, the top 2 centimeters of soil. The open circles are litter samples, everything above the soil layer. And you can see how well they split out. I drew this line in here, but they just split out perfectly in space between soils and litter. And then all the red dots are ones from sites that had just burned, and all the blue dots are from sites that hadn't burned for at least 1 year, but at most 3 years, okay? And what you can see right away is that there's no overlap, right? In the litter, there's no overlap between all the red dots and all the blue dots. And in the soil community, there's a little bit of overlap, but not very much. And so fire distinctly, recent fire distinctly fundamentally changes the soil communities in these fungal communities. And so we know the fire shifted the fungal communities, which is very nice to show, but what did it do for what they can do, their functions? So this is the second part of that experiment where we went out and we took fresh litter and we sterilized it and then we shoved it in these bags, okay? And these bags, the only thing that can get in them is things that are about fungal size and smaller, okay? So bugs can't crawl in there, roots can't get in there, things like that. So really the effects we were seeing were microbial that would differ in these bags. And on the right, what you see is what happened after we went out and picked them up and weighed the stuff that was in them, after 3 months, 6 months, and 9 months. And what happened was that in the burn sites, the microbial community was much less effective at breaking down that litter. This is mass loss. And so unburned sites had about 30% more litter that had been broken down than burn sites. Okay, so the litter in the burn sites would be building up more rapidly than litter in the unburned sites because the beasts that are left after fire are not as good at breaking down the litter. Okay, so serious consequences for trying to manage fire, and that these tiny microbes that nobody can see are having big implications for the buildup of fuels. So that's just an example of some of the research that we work on. But I want to move on and talk to you guys about the platform that I think would be really cool for some of you potentially to use in your classrooms to ask biological questions. And it focuses on these guys called plant endophytes. That term has been thrown around and it's coming to represent things like biodiversity, these catch-all weirdo terms. So now, stricto sensu, or whatever it's supposed to be, plant endophytes are just microbes that live in living plant tissue. Okay, that's the most general term that you can have for them. Okay, but traditionally they were really thought about as fungal generally, and that were asymptomatic. Okay, so that basically you couldn't tell that they were there in the plant for the most part.
All these heterotrophs?
They are all heterotrophs. Yes, yeah, they're all heterotrophs. And that broader definition probably includes things that aren't heterotrophs. But yes, all the ones I'm going to talk about is more that traditional sense about fungal, and mostly asymptomatic, although that's kind of environmentally sensitive as to how asymptomatic they are. But when you think about these guys, this is just some microscopy pictures of what some of them look like. Some of these guys are actually from K-State researchers, so they can be great. You know, you can think about this like the extended phenotype of the plant, right? That just like us that are covered in microbes and have microbes inside of us, that the chemistry that we have in our body, the way that we grow, the responses that we have, our immune system is built not only on our genotype, but on all of those microbes that live in us and on us, right? And it's the same thing here. All of those plant endophytes play a role and play with the plant genotype to create what you see as the phenotype of the plant. So why do we care about them? Well, I think one of the important things is that agriculture, like Peggy talked about, it is essentially dependent on these things. Almost all the plants we know of have mycorrhizae. Many of them have endophytes as well in this traditional sense. Agriculture is dependent on them. In fact, people are using them in agriculture right now. So they add arbuscular mycorrhizae out in the field. They also add just these endophytes that can have impacts. Trichoderma is a good example where people are spreading it and it fights off pathogens in the field. If we understand a little bit more about them, clearly conservation can utilize them as a tool to achieve their goals. I think Peggy gave some great examples of how restoration might be able to benefit from adding soils or particular groups of mycorrhizal fungi. The same could be true for plant endophytes. You know, but I think that there's also maybe an avenue to be made with students that think they're going to be doctors or that are really excited about being doctors, not just at the undergraduate level, but below, and saying, you know, there's a huge number of parallels between what we don't know about ourselves already and what we don't know about these plants and the things that live on them and in them, right? And if you learn more about plants and you don't have to deal with all of the restrictions that come with working with humans or with mammals even, you might learn quite a lot about how those interactions work when immune systems get turned on or off, and how you might be able to utilize those in a system outside of plants. So I think that parallel really seems to resonate with a lot of students. We have two pre-med undergraduates in my lab that we're trying to bring over to the dark side slowly. So I think there is room for that kind of stuff. So what do you do? What does this actually entail? I mean, I think where the rubber hits the road is how hard is this? Like, can I actually do this in my class? So Julie, I'm not gonna put her on too much on the spot. I think we decided like 2 weeks ago when we remembered about this conference, we were like, oh, maybe we can do something for a conference. What can we do? And we were like, I don't know, maybe we can culture some stuff. Okay, how would I do that? So within, I don't know, maybe 2 days, we figured out how to do it, and our classroom did it. Basically, you dig up some roots from whatever kind of samples you want. You stick them in dilute bleach and some ethanol, and then you put those on these plates. We'll play with later if you guys want to. They're just basically boiled potatoes with a starch in them to make them solidify. They're called potato dextrose agar. They're super simple to make. Before I arrived at KU, I already had a graduate student, and she didn't have access to an autoclave, and she made these at home. So you can make them pretty simply and cheaply, but you know, if you got the money, it's nice to be able to buy the pre-made mixy things. We'll talk about, you know, I've taken these also, yeah, I don't want to get aside. So you can culture lots of different isolates simultaneously from those roots, and then if you want to, you can separate out the individual ones and play with those, or you can just say, you know, what did the community look like? So this is what that looks like. Here's the roots. You surface sterilize them and put them on the plates. After a few days, you usually get a few different kinds of things, and then if you want, you can separate them out, and you can have this cool library of different beasts that are microbial, but that now your students can actually see with the naked eye, and that's kind of a huge thing for me. Is you know, so much microbial work or DNA-based work is almost faith-based, right? That there's something going on in it too. That being able to bring it out and make your students see it is really cool for them. Yeah.
So in a high school classroom, if you were doing that, would you at that point just have those plates sealed and just never open those again?
Yeah. So that's the next slide. We're going to talk about that. But
I know when you do like feet and hands.
What was the incubation time on the pictures up there?
I would say most of these have probably been on for about 3 weeks. 3 weeks to a month at most. probably from here to here, especially when you get field routes, it's pretty fast.
It's like
There's
5
some-
days.
Yeah, she's got a ton of stuff on hers already in a week.
And those are just sitting out on a counter or?
Room temperature. Yeah. What was I gonna say? Oh, so I did this in first grade and second grade because I'm the outreach guy for my kids' elementary school. We seal them and we say don't open them. Actually, I'm going to talk about that. So here's a cool experiment just that my graduate student was working on that you guys, I think is a good example. She went out to the reservoirs and she went further and further into the water and she cultured a lot of different things from them. This is what they looked like originally. And then she built a library. And so she can ask questions like, how does the diversity change as you move into the water, as you get more and more inundated? How does the groups of fungi that you have there change? Are they all fast growers or slow growers? Are they snottier? Are they those are the kinds of things. And they make beautiful cultures if you individualize them. So that's a really important point of the students describing them. Is if you don't do DNA techniques on them, you can still say things about them, right? You can say, this is the fluffy, gray, cloudy one, right? And this is the really slow-growing, yucky, yellow one. And this is the stringy white one with the middle volcano-y thing. That's fine, let students describe them however they want to, and then batch them, right? Say, oh, most of these guys look very, very similar, and so we're gonna call all those the same morphotype. It doesn't matter if you're right or not, okay? It doesn't matter if we broke out the DNA techniques or the microscopy, if you were right, it still allows them to bend things, which is important for doing math on them, right? Statistics and stuff. And if you do have access to a microscope, this is not a high-powered, very expensive microscope, but even some of those USB microscopes you can get for like $40 at Best Buy, you can do some pretty good microscopy with things like that. So it is cool if you want to teach them more about how the growth form of these things works, because that's a really valuable thing that fungi actually differ from some other organisms. They don't just grow bigger, they grow apically, so they grow linearly and branch and stuff. So anyways, if you want to get into the microscopy, you can. I'll let you have it. So really, how hard is it? Here's a couple of things that I think. One is that the materials are extraordinarily cheap. There's a huge diversity of potential questions. I'm talking about plant endophytes here, but like I said, in the elementary classrooms, we just let kids stick their face on it or touch it to the water fountain or touch it to the erasers and just see what grows. It's a really great tool for discovery. Everybody got inspired, I think, by biology. And here, a lot of people get inspired by hearing stories about people mostly old white dudes traveling the world in boats and finding stuff. And the beautiful thing about this is that we know very, very little about them. So people can go in their backyard and find new things, like things that people don't know about. That's beautiful. That's discovery, right? And I think that's really inspirational that kids can have that role even, right? There are things like any things that you need to remember that are important lessons to make this work. First is that there is contamination. And by that I mean that you carry things on you and there are things floating in the air that can make this hard, that can make things grow on it when you didn't put stuff on. So we do a lot of control plates. We leave plates open when we're culturing things, and then everything that grew on them and on the other plates is not real. We touch plates ourselves to see what we brought with us because we're usually the dirtiest part that comes in there. There's hazards that need to be explained, just like all experiments. I would say that the biggest is that these things are growing in concentration in a plate. So people that have allergies, if you open it and take a big whiff of it, you're going to get a lot of allergens. These plates aren't designed for culturing human fungal pathogens, and generally, I would say they won't grow well on it. Most of the pathogens that we know about for humans that are fungal. But I think as with anything, as with any physics or chemistry experiment, it's best to just keep them sealed, right? It's best to just be safe. So we generally keep them sealed unless we were going to sub-isolate them, in which case we would probably do that. We sometimes do that in a plexiglass box, but we also sometimes just do it like in an open air, so we don't have to worry about those things. But in general, the major danger, I think, is people that have allergens. You're growing what are essentially molds in high concentrations a lot of times. Okay. Right. So this is what I was just talking about. Julia's already done. She's at this stage, I think, with a bunch of weirdo, funky things growing all simultaneously. They're asking questions about different plants, I think. So they were culturing different plants. So what do you do once you have them? So this is outreach to a high school that we were working with in Austin, that they love to culture themselves and see what grew, then we brought them back. I'm happy to do that with you guys today if you want to touch them or take something outside or whatever. So now that you have all those gross ones like where Julie's at, what can you do with that? So one is that you can compare the communities among sample types, and this is the opportunity for your students to say, well, how do we predict those sample types are going to differ? And they can choose which sample types they're comparing, okay? So the first is to make the sample table. Here's the sample name, and this is just an example of where they might have gotten stuff. They're going to want to have replication because that's kind of an important part of science. So they have 3 different goldenrods, all from roots, all from an old field. Julie collected them all, and she all did her own mistake. And then you might have other plant species. Obviously, that's what you wanted to have those replicated as well if you could. This is just an example of a way that you might cut the cake, right? You might say, I want roots and leaves. I want different plant species. I want all from the same place. You want to take as much data as you can. You might have multiple places like prairies versus old fields, as Peggy was talking about. You can do, you can make up as many different derivations of this as you want to. Then you take the sample names, and for each of those, all you're going to do is take those descriptions and say when you had one in that sample, right? So I had a snotty white one in row 1, and then I had a brown furry fast grow one in one here, right? And so you just create this matrix that says when did I have who, where, okay? And you can take those 2 pieces of data together to compare the sample types in, this is a great example of talking to your students about, well, here's one that's all over the place, a generalist, and here's one that's very rare, So that's a great opportunity to talk about things like that. This is potentially something that everybody has on them or that's in your classroom instead of actually being a ubiquitous endophyte. Okay? So this is the kind of thing you can do. This is much simpler than that fire ordination that I showed you. This is between fungi that we got from native sites, from sites that have been disturbed, or from sites that have been converted to pasture. But it's the same concept. If the points overlap, the communities are similar, are not statistically different. If they don't, then they're not. And you can make these really easily from those 2 tables with free cell. Okay, so if you actually go to the trouble to isolate the individual fungi, you can do cool experiments with them, okay? So the first one is you can make them fight each other, i.e. the Pokemon. They really, especially I think Pokemon's been out long enough and I have enough kids with it, that trapping things and then making them fight each other is really popular. They understand that, they've internalized that as a thing. So this is Stephanie de Leon from Austin, and she basically scanned these plates in Photoshop, I think, or Adobe or something like that. She just used a lasso tool to measure them after a set amount of time. Yeah, sure. Yeah, so you can see this is 6 different treatments. She scanned these with just a regular scanner. In fact, you can see here, it's just an old printer. So she just put a piece of black paper and then scan them, and then she could take that scan and measure them. And students have done that in my undergrad class too, so it's super cheap. And then they can actually have a measured area for how big one is growing when it's fighting another one or not. So another one is grow them in different environments. Undergrads in my class a year ago were somehow able to get another professor to send them chytrid fungus that infects all the amphibians in Sardinia. So they mailed that to our lab, and then they try to grow it on plates like this in different environments. They grew it at different pHs and different water qualities to look at how those things might affect the growth of it. And this is the data that they generated, and these are the number of pixels, which they generated in the same way as Stephanie did. So they could see that at high pHs and at high temperatures, it grew really well. And that lets them think about, well, what's going on in our waterways that's maybe affecting chytrid fungus spreading to our amphibians. But they came up with that idea on their own and actually contacted the professor, and then chytrid showed up on my doorstep. A couple of undergrads also did something with oyster mushrooms. You can buy these kind of things at Home Depot and grow your own oyster mushrooms. Basically, they infected those with they put some oil on those to see if they could eat oil. because they were really interested in what they could eat. I think there's someone talking this afternoon about plastics, microplastics. So you know, see what they can eat, see if they can eat plastics, add the plastics, and see if they grow better or not, or how much is left. There's lots of varieties of different things you can do once you have them isolated to see if you can do a better job. So why would you want to do this? I just took a little bit of time, and I talked a bit with Steve Case. We're trying to, I know some of you guys are graduates of the You Can Teach program. We're trying to build modules so that, you know, you don't have to just do all this willy-nilly or contact me all the time, but it can kind of be a module of things you can do. We're building one off the fire and fungal work. But these are the three life sciences areas that I thought that this kind of work would plug into effectively for EGSS. The first is how do organisms live and grow. That's the structures and processes. You know, you can sit there and watch cell growth and reproduction. You have living models for them eating things and respiring. An example experiment of that would have been, you know, how does pH and temperature affect it? This is that thing that the students came up with. How does that affect their growth and potentially their reproduction? And then in the realm of interactions between organisms, I mean, these are endophytes, so they already live in plants, right? So you can do tons of experiments with reinfecting plants and see how they grow. But thinking about interdependence of organisms, it's not really explicitly in there, but fighting of organisms I'd see as interdependence. I don't think that NGSS wanted to promote fighting as a life science kind of thing, but it is an interdependence thing. The dynamics and resilience of organisms, you know, looking at how they're affecting plant communities is that interdependent stuff. Thinking about shared traits, adaptation and diversity, humans and biodiversity. A couple of examples might be just to get that diversity of organisms, see the ones that are most snotty, and look at them, you know, be like, wow, both of these are snotty. They might not be related, but they both come out snotty. You know, that's kind of a cool thing about convergent evolution. What we're thinking about humans and biodiversity is we shift the kinds of management that we do on landscapes, how that shifts those communities. That's humans and biodiversity. Right, so with that, I just encourage you guys to get dirty, and I appreciate Julie inviting me, and happy to answer any questions. I also have a ton of plates, in fact, more outside. We've done this at the Natural History Museum. You guys are welcome to take some, and we have the seal for them too, the parafilm. You're welcome to touch them yourselves, or take them outside and touch a leaf and see what grows off them. Seal them up, write down what you did. We don't have to do it now since I know we're on a limited time schedule, but I'll have them if you guys want to play with them. Okay.
Are there any questions for Mr. Sykes?
Yeah.
Can you elaborate on how to make the potato auger? If it's short, if it's a
Well,
thing.
Google could probably do a much better job than I can, but potato dextrose auger is basically boiling potatoes, putting them in a cheesecloth is how you get the starch out of the potatoes, and you don't have floating chunks of potatoes in your plates. They usually do it in a pressure cooker, or you can even just, I think you can boil them. We do most of ours in an autoclave, and it takes very little time, and we make lots of them. In fact, we make them all. We did them for breweries, because the brewery wanted a thing like this, and we did it for the Natural History Museum. We've done it for Julie's class. It's not really that hard for us to make a bunch of them at a time, but if you want to make it at home, there's tons of better resources online for how to make them. We actually add a small amount of antibiotics to ours, but so we don't get bacteria. But the pH of the plates and the starch is really not amenable to bacterial growth. So if you do get bacterial growth, it's not very much, even if you don't use antibiotics. You have something you wanted to add?
That's what I was gonna say, is that I feel a little more comfortable using the antibiotics plates, but we haven't opened ours, so I should have brought pictures of them, but I have the data that looks just like what you put there, except agreeing on those descriptions. Like when I went back and looked at it, several different students had different ideas about blue, green, dark green.
Yeah, and you know, that's a great opportunity in biology to show them that science is subjective, right? I mean, it doesn't matter how good your tool is, everybody rarely agrees on the interpretation of that tool. So this is just the outset of that, saying, you know, I don't think that it's green snotty, I think it's green crusty. It looks really crusty to me. And that's okay. Classify things differently, do the analyses two ways. If people really fundamentally disagree about it, see if it changes how much different roots are from leaves, or how much different old fields are from prairies.
Yeah, that's a really great message. During our board meeting, right after the keynote address here, you will have 15 minutes to start eating lunch if you'd like, or you can go outside and
It was.
Okay. You could go outside and do some plain stuff then, and you'd have 15 minutes to just kind of explore. There are also some really great plots just north of here. To go figure out. So let me introduce to you Dr. Ted Morgan. He is a researcher that some of you are already familiar with. He's been involved, especially around the Manhattan School District, New York City School District, with a lot of education outreach programs through a GK-12 program that was there for a long time, funded by the NSF. And then he kind of made relationships with our current president, Noah Bush, and has just kind of stayed involved in certain things. He is an advocate for public education and for diversity and for acceptance of many groups of people who are, I think, underrepresented in our society, unfortunately. So I used to hang around him a lot, thanks to my wife. So I've got a little bit of a prior relationship here and some bias towards Dr. Morgan. But I do respect that about him, that he is very much pro science teachers and just teachers in general. So he's going to talk to us today about building relationships between undergraduate and graduate level researchers, higher education, and us lowly public school teachers. And I am excited to let him talk to you.
All right, thanks, Drew, and thanks for the invitation to this meeting. It's been great, and the thing that I'm going to tell you about is what you've seen already with the 3 talks that preceded mine. So I'm going to talk about the need for university professors and university researchers to interact with people that are in secondary education classrooms. And so this emerges out of work that we've done in my lab over the years, and it also emerges, I was just watching the prior talk, and it emerges because I have an interest in this, so I